Okay, so this is the final one of the key area 2 videos for the structure and replication of DNA key area, and this one is on a process called PCR, and this is an important process that you really need to get your head around. So,
now the idea behind PCR, it's about DNA sampling. Now there are some groups of people who need to be able to create copies of the same DNA. Okay, these groups might include forensic scientists who are investigating a crime scene. So the idea is you've got a tiny sample at a crime scene and you need more of that sample to do your DNA testing on. Genetic researchers, so you've got one person who's got a really rare genetic disease and you cannot be going and stabbing them and collecting skin samples all the time. You need to take one sample and then just keep copying it so you don't really ruin that person's life. Biomedical scientists, again, for similar reasons, they might need samples of DNA just to try various treatments on. If they're developing a drug to cure a disease, they might need large amounts of samples of DNA to try that on. Paternity clinics, so again, if you're looking to find out who's the daddy or the mother possibly as well, if you're looking for genetic testing in general, is that again, you cannot take a massive sample of DNA off a person without them really protesting about it. So instead you take a tiny sample and get the paternity clinic to copy it over and over and then do their test. And medical technicians as well. So say you're worried, do I have a particular genetic condition? Here, take a sample of my skin, go and do some copies of it and then test it for that genetic condition so that I know what's going on.
okay. So for PCR, the main idea of it is it is a process which can amplify DNA, and the reason being that these people who make the sampling that Miss Mills has just mentioned, they need large samples of DNA to be able to do any of the various jobs that they need to do. Because if you just have a small sample of DNA, it is virtually impossible to get enough information from that. You need a large sample of DNA to get appropriate information from it, and that process that we used to do to amplify the DNA, so to make more of the DNA to be used, is this process of PCR that we're going to look at.
Okay. Now, you could say, right, why don't you just grow more cells? So why don't you just take a sample of the cells and, like we've learned with stem cells, feed those cells lots of nutrients and stuff and then grow a large sample of flesh out of that? The problem is there, it's really time consuming. Cells might take, I don't know, a couple of days to do mitosis. It can be really costly. The nutrients that you have to feed your cells to make them grow might be really expensive. And errors happen during mitosis. You know, we know ourselves is sometimes during mitosis, a mutant cell might be made. And so that's not ideal. So instead, another technique called PCR is used to directly copy the DNA from the cell. So instead of needing to copy the cell, and extract the DNA, it takes a tiny sample of DNA and just copies that. The best part is it can be done in a machine as well, which you can see on the little diagram there. It's a really very expensive machine, which is why we don't have them in schools. But it's brilliant. It automates the whole process. It's fantastic.
Okay, so important definition. So your definition from PCR. First of all, the idea that it stands for the polymerase chain reaction, but you can get away with just saying.
You never have to call it the polymerase chain reaction. PCR is always fine.
Yep. So your important definition is PCR is a way to amplify a section of DNA. So those bits in brackets are not important, that's just to give you more information, but the definition PCR is a way to amplify a section of DNA. And this could be anything from a whole chromosome to several genes, or just to one small tiny part of a gene. It can be anything between that, but it's all about amplifying it, so making more copies of that same bit of DNA.
Okay, so the ingredients for PCR look really similar to the ingredients for DNA replication, because that's pretty much what we're doing. We are replicating DNA, but instead of doing it naturally inside a human person, we're doing it in a machine. So it needs to be a little bit different. So thing you need number one is your DNA sample. So that's going to be your template that you're going to create more copies of. You need primers. Okay, so again, these short sequences of nucleotides that are going to kick off the DNA replication, you need those. Now, scientists have gone out into the world and discovered basically how to create primers. It's really clever how they've done this, but they've looked at how to make them artificially, which is brilliant. The third thing you need, now this is an important thing, is you need heat-tolerant DNA polymerase. It's not the same as the DNA polymerase inside us that's copying our own DNA. It does do the same job, but it has to be heat tolerant because during this cycle, temperatures are going to spike and drop and spike and drop. and the DNA polymerase needs to be able to not denature during that time. If it's just normal DNA polymerase that you put into a PCR machine, when the temperature gets to 90 degrees Celsius, that polymerase, as we learned in National 5, that's going to denature, it's not going to work anymore. So it has to be this specific heat-tolerant DNA polymerase, or if you prefer, you can call it Taq polymerase, but I find the heat-tolerant is easier to remember because that's what it's having to do. Okay. We also need free DNA nucleotides. If we've got a DNA sample, we want to build more of it, we're going to need some nucleotides to build the new strand, and you need a PCR machine costing about £10,000, I think, for a standard PCR machine now. It's a lot cheaper than it used to be.
Okay, so now we're going to go through the actual stages of PCR. You need to know all these stages. You need to be able to identify them. And the really important thing, and you can see that it's in bold here, is you need to know the temperature that these stages occur at. Like Miss Mills just said, temperature is going to go up, temperature is going to go down a lot throughout this. And exam questions love to ask you what temperature is this stage done at. So the first stage, obviously, to start off, the DNA is loaded into a PCR machine. You can't do PCR without a PCR machine. And the temperature it goes up to around 95 degrees. The reason we say around is sometimes might say 94.5, 95.5, maybe 96, but we're just going for around 95. If you say 95 degrees in the exam, you're going to get it right. So that's just why it says around, but 95 is the easiest thing to say. And the reason it's going to go up to 95 degrees is because that is a temperature that can cause the hydrogen bonds to break. So like in DNA replication, we needed the DNA to unwind and then the hydrogen bonds to break to free up the bases. Exact same thing happens here. The heat goes up to 95, hydrogen bonds break, and then our bases on our nucleotides are now free to then be replicated from.
Okay, stage 2, the temperature drops. Now this is to allow the primers to bind. If the temperature was still high at 95, the primers wouldn't bother, like hydrogen bonds couldn't form, so the primers would not stay put. So you drop the temperature to around 60 degrees Celsius. That temperature again is an important one that you should know. Primers are added and they're allowed to bound to the 3' ends of the fragment. Now note, the high temperature causes the whole strand of DNA to separate. So it's not the same as standard DNA replication where half of it's still bound together and the bottom end is open. It's not like that anymore. Because the whole strand is at 95 degrees Celsius, the whole strand separates into two basically completely separate strands, each with a 3' and a 5' end that's open. So there is no lagging strand or leading strand on this. They are both going to replicate really nice and continuously. Now the primers are specific sequences of nucleotides that bind to specific areas of the DNA fragment called target areas. That sentence is surprisingly important. Those of you targeting your A's and your B's, is that what are primers comes up as a common exam question, and it's usually worth 2 marks. And the first mark is specific sequences of nucleotides, and the second is binding to specific areas of the DNA fragment. Okay, now the benefit of this is, say you've got a whole chromosome, but you want to copy only the hair color gene. Okay, so you only want thousands of copies of just the hair color gene for blonde. What you could do is find out what are the bases at the start of the hair color gene, and you know they're like A, G, 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 C, C, C. You can make a primer that only matches the start of the hair color gene, so you only copy and bind primers to the start of the hair color gene. If you want to copy the whole chromosome, totally possible too. But this is how we work out, does a person have a specific genetic mutation, is we learn from huge amounts of genetic research what are the bases at the start of the mutated gene, and we make primers to match them.
Okay, so stage 3, again, temperature change. This time it's increased again slightly to around 75 degrees, and this is because it is the optimum temperature for that heat-tolerant DNA polymerase. So again, optimum temperature, we talked about it at Nat 5, it's where it's going to be working, or it's got its highest activity for that enzyme. So this is where the heat-tolerant DNA polymerase enzyme is working at its best, at around that 75 degrees. So the DNA polymerase does a similar job to what it does in the normal replication. It adds in these free nucleotides, and it always does it from that 3' to that 5' direction.
I love the story about how the polymerase was discovered. They actually found bacteria, extremophiles, next to volcanoes where the water is super, super, super hot, and they thought, if these bacteria can replicate and do DNA replication, they must have DNA polymerase that doesn't denature. And they actually extracted the DNA polymerase out of those bacteria living at the bottom of volcanoes. It was amazing. Okay, so stage 4, the strands are complete. So you've got 2 new strands of DNA. So you've gone from 1 fragment of DNA to 2 fragments of DNA. You can keep going through this cycle over and over again until you've got as many fragments as you need. So 1 turns into 2, 2 turns into 4, 4 into 8, 8 into 16. That can come up as an exam question, is something like starting with 2 fragments of DNA after 9 cycles of PCR, how many would you expect to have? So the idea is, again, it's like those mitosis questions that we used to do in National 5, which is if you start with 1 cell, how many cells should be there after 16 divisions? So it's not the 2 times table, it's just doubling those. So 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, and so on and so forth.
Okay, this is just a little GIF. It's just showing you that process of PCR as it is, and you will see the temperatures at the top. Obviously, we said around 95, we're going around 95, we're going around 75, we're going around 50, it's saying 94, it's saying 72. It is around the numbers we're saying. We're saying go for the numbers that we've said, not the ones on this GIF, although they are pretty accurate, it's pretty much the same. But it shows you a very nice clear image of what's actually happening along with the temperatures. So it's a really useful GIF. I would say watch this for a bit, play it over a couple of times to just get used to seeing what is actually going on in this GIF.
Yeah, and in terms of the numbers, I think I took them directly out of the course notes, so those are the ones I think the SQA wants you to know. Okay, now this is a really important picture. There's quite a few exam questions where instead of a picture of the stages of PCR, like stage 1, you've got your 2 fragments that are separating. Instead, they give you a graph that looks like this, and it might be that the temperatures are listed, and it might be the temperatures are not listed. They might highlight a particular area and say what's happening at this temperature. Okay, so it might just be a graph that's tracking the temperature, and they're saying what's happening at 95 degrees, and you need to know, oh, that's the high temperature where the DNA strands are separating. They might ask you, why does the temperature drop to around, in this one, that's about 50 degrees Celsius. And for that one, it's so the primers can attach. Okay, they might ask you, why does it then rise to about 70 degrees Celsius? And that is the optimum temperature for your DNA polymerase. So I fully expect in the exam or in any tests, be aware that you might have to deal with graph questions like this. There's a lot of them in the PCR past paper pack. I really recommend that you try them, okay? It's a really important skill to build up on this. It showed the SQA loves it because it is graph reading, it's knowledge, it's all of those skills bunged together, and they absolutely love it. So it's highly likely to come up if they ask about PCR.
And I'll just point out that where it says primer annealing just means attaching. Sometimes they say annealing, it just means the primer's attaching at this point, just in case any of you were confused by that.
Okay, now these links, you won't be able to click on them because this is just a YouTube video, but to access them, you can go into the SWAY and the links will be available there. Okay, it's just a couple of videos. The top one, I think, is the one that I really like because it does show the process happening over and over and over again. So 1 fragment, and then they separate the 2 new fragments that are made, and then the 4 new fragments, and you get to see that doubling happen really, really easily. Okay, now that's the end of this PCR section. So that's all the basics that you need to be able to know about PCR. A little bit similar to DNA replication, but you need to be aware of the differences between them. Okay, it's really important, those different DNA polymerases, the fact that there's no lagging and leading strand, and also be aware of the uses of PCR. That might come up occasionally, as in who would need to use PCR or what's a function of it.
And that's the end of the second key area of higher human for the first unit. So that's your structure and DNA one done. That's you've got 3 videos. Feel free to go back and watch them as much as you like to try and get your head around it. Make sure you're looking at the SWAY. It's obviously just got these notes in a more accessible thing where you can click on the links with a few more pictures there. Also use your PLP. It is by far the best thing you can have for notes. It's got everything that you need to know in terms of what you should be saying, and obviously matches right in with these PowerPoints and things. And this week you will have your Kahoot as usual. You will have your homeworks. The past papers will be up. One thing we would like to stress, because a lot of you didn't do this very well, is you do not need to hand in your notes. We are trusting that you are handwriting your notes. We're going to ask to see them in August, but we are just trusting you. You don't need to submit pictures. but you do need to simply type the words done and click submit. And the reason for this is it is just so we can track who is doing it. We're trusting that you're doing it. Similarly for the Kahoot, once you put the word done in and submit it, it means we know to go on to Kahoot to go and look at your score so we can then take note of it. So it's for the purpose of us to see who is doing work and keep an eye on you. Even if we're not getting you to submit it, we can go and see your Kahoot scores, and we are trusting that you are doing these notes. The only thing you have to submit an actual file for is the homework. So please make sure you're doing that, because a lot of you, it's going to go down as that you haven't done last week's work because you didn't bother to click submit. And we're fine for this week because it's the first time doing it, but we're not going to go chasing you up every week to find out did you do it or not, or have you just forgotten to click on it.
Okay, so next task that will be up in the next couple of days.